First of all, I would like to thank the CCRI working group as well as the BIPM for letting us have this opportunity to organize our first stakeholder together. It's been quite good 2 days. There's been lots of useful discussions that I think are going to be useful for the AlphaMet project. So I'm going to try to give a very brief overview, very high level of what we're trying to achieve within the AlphaMet project. Obviously, I'm just one person out of a very large consortium. So I'm going to do my best. We have quite a few of the colleagues within the consortium, and we have got all the work package leaders here. So if there is something I don't know during the questions session, of course, everybody can contribute. So you'll see that I highlighted the word metrology here because these projects are very much about characterizing the measurements and trying to improve the measurements, in this case for targeted alpha therapies. And we have seen the traceability pyramid quite a few times today, so I think I don't really need to go into it, but just to highlight again on why metrology is important, especially for nuclear medicine, as it provides an international measurement infrastructure which is stable over time, comparable between locations and coherent. And it achieves that by establishing traceability chains. And I'm not going to give the definition again, but just highlight that here is very important that we calculate uncertainties because we can't really do any comparison, meaningful comparisons within different centers, which is very relevant for these applications, not just targeted alpha therapies, but any type of therapies. We do need uncertainties if we want to do any meaningful comparisons. But of course, the measurement chain in nuclear medicine doesn't just end with measuring your activity on your radionuclide calibrator. We have a very long chain of measurements, which goes from the activity that is either administered to the patient or that you use for doing your corrections or your calibrations or any type of quality control system. Then obviously you do your imaging and you have to process those images. You have reconstruction algorithms. You have a lot of black boxes that you don't know what they're doing. Then you need to do some sort of image analysis, do segmentation, registration, and so on. Then you go into the dosimetry. You need to do a sequential imaging. Again, more measurements that you need to bring together, and then finally do your dosimetry. So as you can see here, this is a very simplified view of the whole measurement chain, but I just wanted to highlight that we really need to understand each of these measurements and have an unbroken chain of calibrations with, of course, again, associated uncertainties. So before going into what we're doing into AlphaMet, I thought it would be useful to look a bit on the history of the different projects that we've done on MRT dosimetry with Uramet. So we've been doing this since 2012. We had 3 projects. The 3 projects have been mainly focused on beta therapies. So we started back in 2012 with the Metro MRT project. At that point, I was not working in our Metrology Institute. I was working on the other side. And I do remember when MPL came to the hospital to try to discuss about this project. And I remember I was sitting there as a postdoc and they were asking me, so all of these dosimetry calculations that you're doing, what sort of uncertainties did you think you have? And I was like, well, I don't know, but if I had to guess, I don't know, 50, 80% probably. And they were like their faces sort of just used to the 2-3% uncertainties on like half-lives and things like that. So I think that first project, it was sort of about trying to learn to talk to each other. And it was quite interesting. So then we moved into the follow-up project, the MRT dosimetry project. And then finally, we had a smaller project, which was about support for impact in terms of trying to bring the results of the previous projects closer to the clinic. And I just wanted to highlight a few, just basically 3 examples, basically from the MRT dosimetry project, which I think had quite a good impact. So we actually contributed to the publication of the first international guidelines for doing uncertainty calculations for MRT dosimetry. together with the ENM. Of course, there is always more room for research, so there is always improvements. And I think this probably will require some sort of update when we are talking about targeted alpha therapy, because obviously there are new challenges that need to be considered. Another interesting result from the MRT Symmetry project is that we perform a multi-center international comparison exercise of lutetium 177 SPECT-CT imaging. And here we were looking at the quantitative accuracy using various phantoms. And at the end as well, we had a validation phantom that was based on a 3D printed organ phantom. And just picked up a couple of results here, and you can see the measured activity with the imaging versus the true activity. And for the kidney and spleen, we can see some variations where we are sort of close to being able to harmonize this. And the good news is that these results are now being used to guide a new L accreditation for quantitative SPECT imaging. So the goal here is to try to improve accuracy and reproducibility for multi-center studies. And on this accreditation, there is an emphasis as well on trying to have traceability to primary standards for activity measurements. And this links back to Johannes's question earlier about the certificates and how we can get the manufacturers to provide that traceability to the end user. So then we can ask the people that contribute to this accreditation, how do we check whether they're really traceable or not? And then the last example that I wanted to bring up is that obviously we're also working towards quality assurance of the Symmetry software. And for that, we created a reference data set, again, based on a 3D printed phantom based on the ICRP 110 model that we used to fill with activities that were based on a pharmacokinetic model that was realistic for a lutetium dotatate therapy. And we used that reference data set to then perform an instant comparison exercise of the Symmetry software, both commercial and research here. And you can see some of the results comparing the calculated absorbed doses and the true absorbed doses. And you can see that in general, there is quite a few differences, let's say. And there is a general underestimation. And you can see some, worrying points, which are quite large differences, even for a spleen, which is quite a large organ. So of course, as I said, this was all beta therapies. We wanted to carry on having more projects and providing more metrology support for MRT. So what was the obvious step was to move into alpha therapies. So obviously, radium 223 is the first and only alpha emitter that is approved for therapies. And because of the success on the ALSYMCA trial improving survival, obviously there's been a huge investment into other alpha therapies, and there is like a global market that keeps increasing. So there's a lot of new therapies based on other alpha emitters that are being considered at the moment. And I just picked up this picture from the new ENM first in human studies, which shows how the number of studies is rapidly increasing in the last few years. And I highlighted here the three alpha emitters, which are mainly being considered within the AlphaMet project. However, what's the problem? Well, we don't really have the robust radioactivity standards for all of these alpha emitters. We don't really have good practice guides on how to do activity quantification, how to do dosimetry. And at the moment, we don't really have the traceability. We don't know what the uncertainties of doing all of these measurements are. And we don't know what the accuracy of whether these measurements can even be reproducible. And of course, we know that we should be doing dosimetry. We've got the BSS directive. So this is how AlphaMet came into reality. We put a bid together. We were successful within the new Metrology partnership program of Euramet, which is co-funded by the European Union. We have quite a large consortium. We have 8 metrology institutes across Europe, and it's being coordinated by CMI, and Jan here is the coordinator. And we also have a good mix of hospitals as well. We have 8 clinical and research centers with 1 affiliated entity as well, because it's obviously very important that the metrology works closer with the hospitals. And it's a 3-year project. We started last September, so we're still sort of starting up, as you will see. So we have 4 technical work packages. Just very quickly, we go from trying to do end-to-end metrology. So we start with the reactivity standards, then we move into the activity quantification, then into absurd dose quantification. And at the end, we also focus specifically on the bone marrow as one of the main organs at risk. So I thought it would be very useful, I think, to go through each of the work packages and see what the challenges are and how we're trying to address this within the project. So if we start with the work package 1 on standards, here one of the challenges is that, of course, as we've seen during this workshop, equivalence is very important because just a single primer standard and a single standard is not good enough. So even within the Nuclear Medicine Institute, NMIs, we have to compare within each other. And yeah, everyone knows the example from Brilliant 223, where the first NMI did some measurements and that was used by Bayer. But then 5 years later, there was a revision of these standards, and it was found that there was a 9% bias. And that meant that Bayer had to change the label from what was before 50 kilobecquerels per kilogram, that changed to 55 kilobecquerels per kilogram, as indeed the patients were being injected with 9% more activity that was originally intended. And the reality is we don't really have fully validated across different NMI standards for other alpha emitters. As well, one of the challenges is that there is international guidance that recommends that the activity that is injected into the patient should be accurate within plus minus 5% for therapeutic radionuclides. And the thing is that we don't really know how well hospitals can actually measure these activities with the radionuclide calibrators. And we know that we've got a lot of challenges with alpha emitters. Again, Denis already showed this paper this morning. It goes through all the alpha emitters and the challenges on measuring this. So how are we addressing this within the project? So we are developing new primary and secondary standards for actinium, lead, and astatine. For each of them, we have quite a few of the NMIs doing the standards, and I highlighted in bold, even though I believe this might be changing, the NMIs that will be comparing within each other, and all of these will be submitted to the SIR as well for establishing equivalence. And then with respect to the second challenge, we are also going to perform an intercomparison exercise of activity measurement capabilities using radionuclide calibrators and gamma counters within hospitals and as well preclinical research centers, with the idea of assessing how well they can actually measure it. And then we will calibrate it with the new primary standards and reassess what the differences are. And here we will try to focus on actinium-225. And the idea is that hopefully we can see some improvements. Here's an example. It's not an alpha emitter, but it's also difficult to measure radionuclide, iodine-123. This is a comparison that was run in the UK, and we can see that when we did it the first time in 1996, and then we repeated in 2015, there was quite a huge improvement in the measurement capabilities at hospitals. And that was basically due to the introduction of new measurement services. So we hope that we will see something very similar for the alpha emitters. Moving into work package 2 and activity quantification. Here, we know that quantitative SPECT imaging is not well established for alpha therapies, and probably not even planar imaging is well established. However, we do need it if we want to do treatment verification. However, again, we have all the problems to do with the low activities that are administered. So obviously, we get low counts, poor resolution, and we don't really know how accurate or reproducible or the uncertainties of these images. So are they actually the big questions? Who will be doing? Is it useful to be doing quantitative SPECT for alpha emitters? And I just thought it would be fun to look at some images. I try to pick an example for each of the isotopes. If I've not picked up your paper, please don't be offended. It's just some example. So for radium here, we can see the planar images compared with the technetium and F18 fluoride, and judge yourself. I'm not going to make any comments. As well for thorium and radium, there's been the group in Sweden doing quite a lot of work, trying to differentiate using different energy windows between the thorium and the radium. But the conclusion was that it wasn't good enough to be able to differentiate both isotopes. Actinium, we've seen already some images. This is the work from Munich, where they did manage to do a SPECT-CT, but obviously they did work on improving the image reconstruction. So that's why the images start looking better. Another one from Katrobil, which again, what can you do with that image? Is it quantitatively good enough? Same thing for bismuth, very noisy images. And then astatine, again, very similar, planar imaging, very blurry, not a lot of statistics. Very similar for lead. This is already a study from 2014. There's been some more recent work from the work at Oslo for both radium and lead. And they actually have looked at the quantification accuracy using phantoms, and is achievable. However, again, these are studies from a single institution, so we've got to take the results with some caution, and it's good to repeat these measurements. So what we are doing within the project is firstly to assess the feasibility and practicality of doing SPECT in terms of calibration, how to do the limits of detectability, how low we can go, how small we can go, whether we need to change the quality control procedures. And again, the aim is to focus on actinium, but we are also considering lead and astatine within here. And Leuven has done already some imaging with actinium. These are their images. And the idea is to try to improve the quantification by using Monte Carlo simulations to generate realistic data sets, and then work on trying out different reconstruction methods to try and improve the quantification. But I think the most important part for everybody will be that we are organizing another international multi-center comparison exercise. Again, assessing accuracy, reproducibility, and quantifying the uncertainties. And we're going to focus on actinium. And we're going to consider, obviously, the three major manufacturers. And the idea is to then propose some guidance to look at whether we can then do another harmonization accreditation for alpha emitters if we find that, obviously, they're good enough for the quantification. Moving to work package 3, we go into the symmetry. We've already seen that obviously high energy, short range alpha particles are impossible to image. So obviously we need to know the microscopic uptake distribution if we do want to go and do the symmetry. And yeah, to our knowledge, the only publication that has attempted to some uncertainties was the one by Nicolas Choin, who is here as well. And he's seen that the uncertainties can be up to 75%. So there is a lot of work that needs to be done in there. And again, usual challenges in homogeneous distribution, complex decay schemes in growth, the daughters translocating. So how do we go from this routine clinical macro dosimetry into the micro dosimetry and vice versa? What is the impact of all the assumptions if we do macro dosimetry. And people are doing macro dosimetry, and I picked up a couple of examples from 2 places that did radium dosimetry. And again, these are 2 different administration activities. However, it's very difficult to compare the absolute doses from the 2 studies, because even the way of reporting it is different. The calculation methods was different, and even within the second publication, they didn't consider the absolute doses from the daughters, which is missing 75% of the absolute dose, more or less. So it's very difficult to compare any results. So we need to provide this guidance. So what we're doing in the project is, firstly, we're going to look into autoradiography and trying to understand what uncertainties are in there and provide recommendations to improve it. We've seen this morning from Brian Miller's talk that there are a lot of challenges in there. We are also going to look into pharmacokinetic modeling and quantifying the uncertainties by using a sensitivity analysis method. So the idea is that by varying the input parameters, we can see how the output varies. And we will be able to identify what are the major parameters that can impact the dosimetry outcomes. And for that, we will be using data from Astatine 211 coming from Gothenburg. And finally, we will investigate the impact of the macro to micro dosimetry as well. And finally, the end is to provide end-to-end guidance on how to achieve end-to-end metrology from the activity quantification to the absolute dose quantification. Last but not least, work package 4 is focused on the bone marrow. And here, of course, we all know that the red marrow is the most radiosensitive organ, and it often can limit the amount of activity that is administered for a given therapy. The red marrow is within the spongosa, and this is composed by the red marrow, the yellow marrow, and the trabeculae. We know that the composition is dynamic. It changes with the patient age, and it changes within different bone sites. And this information is not really currently used for the dosimetric models, for example, based on the ICRP. So there's been some modeling studies where they actually try to quantify the errors by considering what would those factors be if we account for different marrow cellularities. And for alpha emitters, the self-irradiation dose factors can actually have errors of 200 to 300%. So we were doing in the project, we are designing a bespoke bone site phantom, sorry. And for that, we've been looking at trying to collaborate with this company called CPI UK, which have got new technology looking at anatomical printing with some novel materials. But of course, we are finding some challenges because we need to replicate the realistic densities of the different bone tissues. So it's something that we're working on. And the idea is that that phantom will be part of the imaging comparison phantom from work package 2 as well. And then the next step is to try to characterize the spongosa. We're going to be looking at doing MRI imaging and dual energy CT imaging, and to optimize these protocols. UKW have got a lot of experience on this. And of course, we will focus on uncertainty budgets within it. And then additionally as well, using this imaging data from the bone phantom, together with Monte Carlo simulations, we're going to try to assess the image space error from doing the bone marrow dosimetry using these blurry images that we get from the SPECT compared with the real doses. And as well, we will investigate the influence of the geometry of bone metastases on the absolute doses to red marrow by simulating various geometries and looking at the impact of the absolute doses to the marrow. But of course, not everything is the technical work. It's very important that we create impact. And for that, we need to work with all the stakeholders, whether that's scientific associations like IAEA, ENM, IFOMP, and everybody, but as well the pharmaceutical companies, the scanner manufacturers, and all the hospitals as well. I apologize if you already signed up to work on the project and your logo is not here. These are the people that supported us during the bidding process. But of course, this brings me to how you can get involved. Of course, you can look at our project website or LinkedIn page, but don't do it anytime soon because it's not quite ready yet. So maybe give us 2-3 weeks. You can just get in touch either with myself, with Jan, or with anybody within the consortium. You can become an official collaborator, and you can sign up to collaborate in any of the activities that you heard. It will also give you access to attend project meetings and sort of early access to the ongoing research. And it's very easy. It's a very simple agreement. You just pass it to your legal department, probably wait 3 months, and eventually you can just sign up. You can join our stakeholder workshops. We're here on the first one, but we have 2 more to organize, probably summer 2025, and then at the end of the project. To be confirmed where, how, and but if you want to get involved, get in touch. And then coming up, which I think is very interesting, is that within work package 1, we're organizing a survey to look at what the uses are of alpha emitters at the moment in terms of doing the activity measurements with the radionuclide calibrators, gamma counters, but also how people do imaging and whether they do imaging and whether they do dosimetry. And the idea of the survey as well is that we're going to identify the people that will participate in the different intercomparison exercises. So that will hopefully come up in September, and we will be promoting it at the ENM in Hamburg as well. If you're there, come and speak to us. And with that, I would like to thank all the people working in the project. I am sure I have not included everybody in here, so I apologize if I missed you. And thank you for listening.